us know about the bedside monitor. This is the bedside monitor. We use bedside monitor in order to measure physiological signals such as ECG, SpO2, temperature, and also invasive and non-invasive blood pressure. Physiological signals are collected from the electrode from the human body. So these are in millivolts. In order to amplify them, we need amplification. So we use instrumentation amplifier. And then signal conditioning in order to remove the noise artifact. The signals are converted into electrical signals by the transducer. Now these are analog, so in order to convert them into digital, we use ADC, and the digitalized signal is sent into the microprocessor and displayed. Electrodes in order to measure these physiological signals like ECG electrodes and pulse oximeter, blood pressure cuff, and temperature probe. About the electrocardiograph. So, electrocardiograph is the measure of electrical activity of the heart. The electrodes are connected to our body, converts these signals into electrical voltages. These electrical voltages is in millivolts. So, we have to amplify them. And the noise is also filtered out. The bandwidth of the ECG is 0.5 Hz to 150 Hz. So, we use band pass filters. So, in order to remove the power line interface 50 Hz, we have to use the notch filter, and then the ECG signal is displayed. We have two types of leads, unipolar lead and bipolar lead. In unipolar lead, we have lead 1, lead 2, lead 3. It is the difference of single positive electrode and negative electrode. In bipolar lead, we have AVI, AVF and AVR, augmented limb lead. And we have six test electrodes V1, V2, V3, V4, V5, V6. And here a single positive electrode is used with a reference of combination of other electrodes as negative electrode. Let us know about the blood pressure. What is blood pressure? It is the pressure of the circulating blood which is exerted against the walls of blood vessels. Manually we use oscillator method. In bedside monitor we use oscillometric method. So when we on this button, the pressure cuff starts inflating. The point at which the blood becomes constant, it measures the pressure sensor in the blood pressure cuff will measure the pressure, that is systolic, and it will deflate the cuff. The point at which the flow starts is the diastole. So this is how it measures the blood pressure. Let us know how to measure the temperature. So we have a thermistor or a thermocouple. We use this to measure the temperature. So the thermocouple is highly sensitive. So even small change of temperature can be detected by this. So this is how we measure the temperature. And let us know about the pulse oximeter. Pulse oximeter is a device in order to measure the oxygen saturation in blood. How do we measure this? We use a peripheral device such as this pulse oximeter placed to the finger. In this we have LED and a photodetector. LED sends red and infrared lights, and these are sent into the blood. And we have a photodetector. So the blood absorbs infrared and red light in different ranges. Hemoglobin absorbs more infrared light. So through this absorbance, we measure the pulse, like oxygen, saturated oxygen in blood. Use this knob, like we can move it. So we can select by pressing this button, and this is to inflate the pressure cuff, and all the electrodes are connected here. We can see ECG, SpO2, temperature, NIBP. During treatment, it is highly important to measure all the physiological signals. So that is why we call it as a critical monitoring system. So it is highly important during treatment. The electrodes are connected to the patient using electrode gel. Here we can see the ECG waveform, respiration, plethysmograph, and pulse rate, BP, and temperature. If the waveform is incorrect, the patient monitor will give us alarm. Here we can see an alarm.